So when we left off, we were talking about enzymes. And we finished by drawing a picture of the activation energy barrier for a chemical reaction. So we saw that we have reactants with some amount of energy in them. And in order to move forward with our reaction, in order to get to our products, which may have lower energy, we still need to overcome something called the activation energy barrier, which usually without an enzyme, we'd kind of have to wait for us to overcome this barrier. But the point of an enzyme is that it speeds up a reaction by lowering the activation energy, which means that the reaction happens more easily, which in practical terms means it happens more quickly. So that's really the point of our enzymes, is to speed up our reactions so that we can keep everything moving at a steady pace. Now, the way our enzymes work is by basically locking on to our reactants and putting them in a position that allows this reaction to happen more easily. Okay, so the reason they lock on, right, the reason an enzyme has a shape is it's a type of protein. And so it has basically a little pocket for our reactants. Now, there are two models of how this could work. The model you may have talked about in high school, unless they've updated high school textbooks, is the lock and key model. So the lock and key model says that you have an enzyme, it has a very specific sort of lock, and then your reactant joins into that lock like a key. But we saw from our equation of these chemical reactions that our chemical reactions are reversible. So most reactions that we'll see in the body that are catalyzed by an enzyme can also work in the reverse. So we can take those products and convert them back into the reactants. So if we had a lock and key model where our lock is a very, very specific shape just for those original reactants, then this wouldn't really work because those products shouldn't fit in if we just have that one lock shape. So a slightly more accurate theory of how an enzyme works is something called the induced fit model. So the induced fit model says that we have this area of the enzyme. The area where our reactants are going to bind in is called the active site. So the active site is where this is all going down. So it says that this active site, when it binds onto the reactants, that active site isn't a perfect fit. What actually happens is that that active site slightly changes shape in order to fit the reactants, which is why certain enzymes can catalyze several different types of reactions. So one thing may fit super well in there, but other things can also sort of fit. The enzyme can shift a little to allow them to fit. Okay, so in this induced fit model, this is looking at a substrate, S, so that's the same thing as our reactant. It's looking this enzyme E here. So we can see here that the active site in this picture of this enzyme, we have this kind of square active site. Our substrate is round, but this enzyme does accept this substrate. So via the induced fit model, this active site slightly changes shape. So this active site slightly changed shape to allow this reactant, this substrate, to bind on. And then our reaction happens, the reaction we were thinking about, and we get our product. The product gets released. This enzyme goes back to that original shape. So that's the induced fit model. Basically, it's saying that the active site is slightly variable, can slightly change shape, versus the lock and key model, where we'd have just one fixed shape for that active site. So that's the difference between those two things. And this allows us to use the same enzyme to bind on chemicals of slightly different shapes, which allows for our reactions to be reversible because this product could bind back into this site, and then the site would change shape as well and allow us to run this in the reverse. Okay. Another important feature of how enzymes work is that often we're going to need other little pieces to allow enzymes to function appropriately. So we have these things called cofactors and coenzymes. Our enzyme is a protein, but our cofactors and coenzymes are not proteins. So there's something else. So our cofactors are often going to be trace metals, so things like magnesium, things like iron. And they're important components for how the enzyme works. So they'll like bind in with the enzyme or be used by the enzyme in order to allow the reaction that it's catalyzing to happen. Coenzymes work kind of similarly. So the coenzymes themselves don't have catalytic activity. So a coenzyme isn't doing that same exact job as the enzyme. It's not lowering the activation energy barrier on its own, but it's necessary for the enzyme to work the way it works. So our coenzymes do participate directly in enzyme reactions. And we'll see an example of this as we move into metabolism. So our coenzymes are often derived from vitamins, and they'll transfer small chemical groups during chemical reactions as they're working together with our enzymes. So cofactors and coenzymes are like helpers. So one of the things we want to concentrate on in physiology is enzyme reaction rates, because how fast reactions are happening really affects how the body works, so how our different organ systems are working. So the reaction rate, even when it's catalyzed by an enzyme, can vary. So our enzyme's catalytic rate is how many molecules it's generating per unit of time. And this is going to be dependent on the concentration of the substrate, so how available those reactants are. It's going to vary by enzyme concentration. Sorry, okay, someone's joining us late, just got an email. Okay, so the availability of that enzyme also matters. So we need enough enzyme to allow our reaction to happen fast enough. If we don't have enough enzyme, this reaction will run slower. And it's also going to be affected by the affinity of the enzyme for the substrate. So in that induced fit model that we were talking about, the enzyme had a shape. The active site could slightly change shape in order to bind to our substrate, our reactant. But not all things are going to bind in equally well to any given enzyme. And that's what we call affinity. So something with high affinity for an enzyme, it binds in really well. It fits super well into that active site. But something with low affinity for an enzyme, it means that it can work together with that enzyme, but it's harder. The shapes aren't quite right. So that enzyme will have to work harder in order to catalyze that reaction. And we'll take a look in graphical form of how that looks when we're thinking about our reaction rates. Okay. Temperature and pH are also important to the function of enzymes. Basically, higher temperature often makes things run faster, because as the molecules are moving around more, which happens at higher temperatures, they basically have more energy, so they get closer to that activation energy barrier just because of the temperature on their own. Okay, so here we're going to take a look at what substrate affinity means for the reaction rate. So I'm just going to write on with the annotation tool instead of switching cameras, I hope. Where's the annotation tool? There we go. Okay. So for an enzyme and substrate with low affinity for each other, so an enzyme that's not quite the right shape for that substrate, a substrate that's not quite matching into that active site, basically what happens is that we start out, and at the beginning of this reaction, when we're at low substrate concentration, so we don't have very much substrate, because there's not very much substrate in the environment, and the enzyme and the substrate don't like each other all that much, the reaction rate is slow. But as we increase the substrate concentration, so as there's more and more and more of that substrate, basically what we're doing is raising the probability that any given molecule of that substrate will bump into an enzyme and happen to bind into that active site. So the more substrate there is in the environment, the higher the substrate concentration is, the better these two things, or the more likely it is that an enzyme and a substrate are going to bind together, which means that our reaction rate goes up. Okay, so this is what's happening with our low affinity. Okay, I'm going to change colors and draw on the high affinity now so that you can kind of appreciate the difference. So if we have an enzyme and substrate with high affinity, so they really like each other, basically they attract each other, then even at very low concentrations, because they work together really well, if we have high affinity between our substrate and our enzyme, we're immediately going to get that enzyme binding onto the substrate. So if we have high affinity, the reaction rate is higher at much lower concentrations. Now, these two lines are going to come together kind of near the end because we will hit something called enzyme saturation, which is where at a certain concentration of our substrate, we'll be using up all the enzyme that is present in the environment. So we'll eventually level out to a similar level. Okay. So this is showing us how the affinity of an enzyme for a substrate affects the reaction rate. Okay. So beyond how well our enzyme and our substrate fit together, the body also is able to regulate enzyme activity. So the goal, overarching goal here, is for our reactions to run at a steady pace. We don't want our metabolic reactions, for example, to sort of stop and start jerkily. We want to run our chemical pathways in the body very smoothly. So we want to have the right amounts of each of our reactants and products at the right times. And so in order to do that, we're going to see there are various mechanisms and various feedback loops that allow us to slow down or speed up different parts in these different metabolic pathways. And they work via a couple different ways. So first we're going to talk about allosteric and covalent regulation, which has to do with actually basically kind of the shape of the enzyme. And then we'll talk about feedback and feed forward, feedback inhibition and feed forward activation, which are kind of like negative feedback and positive feedback, but more specifically for our enzymes here. Okay. So in allosteric regulation, allo means other. So in allosteric regulation, we have something called a modulator, so some other chemical that has an effect on the enzyme. And it has this effect on the enzyme by binding into something called the regulatory site. So our reactant, our substrate, binds into the active site, but we have another site somewhere else called this regulatory site. So this is why it's allosteric, is because it's this other spot. So it's binding in somewhere other than the active site. And so when we have this modulator bind into the regulatory site during allosteric regulation, we can see in this simplified diagram that it's changed the shape of the active site. So here, the fact that we had this different chemical bind onto the enzyme in this regulatory site is affecting the active site of that enzyme, which means it's also going to affect the activity of that enzyme. So when we change the shape of this active site dramatically, even though through induced fit, we would expect it under normal function to change slightly, when we change it using some other chemical in the regulatory site, this can change basically the affinity of the enzyme for various substrates. So if this change for example, lowers the affinity of our enzyme for a substrate, that's going to decrease the speed of whatever reaction we're running. So that's one way that we can regulate how fast a reaction is going. We can basically like kind of break the enzyme. Not permanently break it, but make it work less well. Okay. So you might see a graph like this. There are both allosteric activators and inhibitors. So an allosteric inhibitor makes an enzyme bind onto its substrate less well. So it makes the enzyme work slower. It's going to slow down the reaction rate. And with an allosteric activator, so an activator basically speeds it up. So an allosteric activator would increase the affinity of the enzyme for the substrate and have the effect of basically speeding up the reactions. So we can see those two curves that we drew basically for affinity before. We can see them here as they relate to our allosteric inhibitors and activators. So we can see here we have this lower kind of S curve with our allosteric inhibitor. So it's going to take more substrate to get the same reaction rate. And the reason for that is this allosteric inhibitor has lowered the affinity of that enzyme for that substrate. With an allosteric activator, we don't need that much substrate to get a really good reaction going, because this allosteric activator has made that enzyme have higher affinity for that substrate. So this is how an allosteric inhibitor activator can regulate the rate of a reaction. So it changes the shape of the active site, which changes the affinity of the enzyme for the substrate. Okay. Another type of regulation that we can see is covalent regulation. So covalent regulation works similarly to our allosteric regulation, but it's a little more specific. So in covalent regulation, we'll have a chemical group that covalently bonds onto the enzyme. So the fact that it's covalently bonding into the enzyme means that it's almost becoming like a permanent part of the enzyme. So this is going to have a much sort of longer term effect on that enzyme, because then we're going to need a secondary enzyme to either help that chemical group bind on or to help that chemical group pop off. So with our allosteric regulation, we had things just forming these sort of temporary associations with our enzyme by the regulatory site. But in our covalent regulation, we're actually covalently bonding something onto the enzyme, which is a longer term change. So here we see in this image, we have enzyme A helping this chemical group to covalently bond onto our blue enzyme. And then in order to get this chemical group off, in order to return this original enzyme to its normal activity, we're going to need to use a third enzyme in order to break that bond. Okay, so during covalent regulation, basically in this course, we're just going to see two types, and they both have to do with phosphate groups. So the two types we're basically going to see is that we'll have some reactions where we're going to bind on some phosphorus to our enzyme. So here we see enzyme plus a phosphate group that's just floating around. And then we're going to have this secondary enzyme called the protein kinase that binds on that phosphate group. So I'm going to flip back to the previous slide just to show you what that equation is for. So that's for this top part. So our kinase here, our protein kinase, is this enzyme A. Okay, so this is showing us binding that phosphate group onto our enzyme, forming covalent regulation, because now we can see we have a covalent bond between our enzyme and the phosphate. And then in order to remove the phosphate from this enzyme, so in order to subtract it, we need to use another enzyme. So this is, flipping back, this is enzyme B. So phosphatase breaks this covalent bond between our enzyme and the phosphate group, freeing it and returning our enzyme to normal function. Okay, so now we're going to talk about feedback loops. That's the sort of nitty-gritty about how we're changing the rates of reaction, the function of the enzyme, by changing its affinity for its substrate. But we also want to think about where in a pathway of chemical reactions this is happening. So usually we're not just thinking about one chemical reaction, so one set of reactants that immediately leads to a set of products. In physiology, we have steps, basically, right? Like this is all like dominoes. You have the first chemical reaction that sets off a second chemical reaction that sets off a third chemical reaction. And that's how we finally get to our end product. And we'll see that as we go through metabolism, right? We don't just have one immediate chemical reaction that happens one step and we're good. We see that there are a ton of steps as we do cellular respiration. So we can regulate the activity of different steps using feedback inhibition or, never mind, I thought it was the next slide, or feed forward activation. We'll get there when we get there. Okay, so what we see here in this diagram is that we have a couple of steps in this reaction that we're talking about. First, we convert A to B, B to C, and then C to D. So what we want to have happen in our cell is we want to very smoothly go A, B, C, D, which means we'd want E1, E2, and E3 all functioning like kind of similarly. We want the rates of these three reactions to be similar because we want to have the right amount of B, the right amount of C. We want this to not stop and start. We want it to just kind of flow. So what we're looking at here, we're seeing that E1 and E3 have these specific rates, but the rate of this middle step, the rate of turning B into C, is dependent on how much of this molecule C we have. And the reason for that is that we have this negative feedback loop from C, where C inhibits this enzyme E2. So it basically goes backwards and it slows down this enzyme. So it's making this enzyme have less affinity for substrate B, for example. So it's slowing down this reaction. So whenever we have a lot of C hanging around, it slows down the step before it because we want to lower the concentration of C. We want to keep the amount of C constant, and we want to keep it constant at the rate that this third enzyme, E3, can run this C to D reaction. We don't want to end up with a buildup of C because that's how we end up with this sort of stop and start, this not steady flow of our overall pathway. So here in this diagram, B to C, this middle step, is going to be our rate limiting step. So if we turn off B to C, if we were to turn it off completely, we can't convert C to D because we needed to go from B to C in order to have any C to convert. Okay, so our goal of this regulation is to proceed at a smooth, steady pace. So we want to think for a second about what would happen if the rate of C to D were really high. So like if C is really quickly being turned into this molecule D, what's going to happen here in the middle with this negative feedback loop? Okay. So if this rate of C to D is really high, and as soon as we have any of molecule C, we immediately convert it to D, this means that there's not much of this molecule C hanging around, which means that molecule C is not really inhibiting this B to C step. So B to C is going quickly. So this basically has the effect of creating more and more C to fuel this really fast step of C to D. This would also work in the reverse. So if our reaction C to D is really slow, that's when we're going to turn on this pathway. So if it's hard to convert C to D and we end up with a ton of C just floating around in our environment, that C that's floating around is going to go affect this enzyme E2. It's going to kind of turn it off. It's going to lower its affinity for B, which is going to slow down this step, which means then we have time to use up the C and convert it to D. So in this diagram, C is an allosteric inhibitor of enzyme E2. And our overall goal is to just have this reaction run smoothly. Okay. So feedback inhibition doesn't always have to work just on that central step. We'll see feedback inhibition also work on whole pathways. So if we go A, B, C, D, sometimes our final end product will affect our initial reaction. And this is basically how we can turn off an entire pathway instead of just slowing it down and making it run at a steady pace. So sometimes we'll want to do a process at certain times, but then when we have enough of our final product, we want to quit making it. And so this is how we would do that. So we could see a picture here of how this could work via allosteric regulation, right? So here's our reaction, slightly different one, but we have one enzyme converting here. We have another enzyme working here. Then we have our final end product, which is this yellow square. And this yellow square is an allosteric inhibitor here for our initial enzyme. So it's an allosteric inhibitor, binds into this regulatory site. So now our substrate doesn't fit very well anymore. So it's lowering the affinity. So it turns off this thing. So basically when this end product, when there's enough of it, it inhibits its own creation. So this is a negative feedback loop. Feed-forward activation is rare. So we do occasionally see that sometimes the creation of a certain molecule actually speeds up some part of the process later on. So in this case, we see a diagram of B acting as an allosteric activator of this final step of this enzyme E3. So this would speed up the C to D reaction here. So we're not going to see that very often in physiology, but it is possible. So that's feed-forward activation. So we can think of it as analogous to positive feedback, kind of, right? Because we're speeding it up. Okay, I want to move directly into metabolism, so I don't have to give you guys any videos to watch over the weekend because I'm sure you have endless videos from all your classes. So we're going to get rolling on this. Hopefully you kind of remember this stuff from chemistry and from your previous courses. So our super basic goal, like what we want to know for this course is we want to know basically how we make ATP, because we want to think about ATP's role in functions of the body. And we're going to be studying the musculoskeletal system at some point during A&P1. So this will importantly really come back then. We'll see how ATP is used to make your muscles contract and allow your body to actually move and do things. So that's why we have to talk about metabolism now. I mean, ATP does tons of important things, but that's the most sort of intuitive link to the function of your body. So we want to understand the role of ATP in energy metabolism. So very, very important. Please, please, please know which parts, which of these steps make ATP and relatively how much they make, whether it's more ATP or only a little bit of ATP. We're going to talk about the different steps kind of broadly, and we're going to group them into glycolysis, the Krebs cycle, and oxidative phosphorylation. So I know depending on where you learned about metabolism, the steps get broken down in slightly different ways, but that's how we're thinking of them here. Glycolysis, Krebs cycle, oxidative phosphorylation. Then we're going to talk for a very brief moment about how we can get energy from things that are not glucose. So we'll talk about breaking down fats, proteins, glycogen, and how we can store energy in these different compounds. Okay. So our main focus when we're talking in this metabolism unit, our main focus is aerobic respiration, which is the conversion of glucose to ATP, to energy, through glucose oxidation. So we have a central reaction that basically describes this whole process, even though we have a lot of substeps. So this would be our original reactants, and then we have like our end products at the very end of the process, even though we'll see that there's a lot of stuff happening in the middle here. So here we have our glucose, C6H12O6. Here we have some oxidation, right? We're doing glucose oxidation. Oh, sorry, we have some oxygen because we're doing glucose oxidation. And then we get out carbon dioxide, water, and energy at the end of this process. Okay. So we can diagram this in different ways. So we can see that we're breaking down our glucose, adding in oxidation, adding in oxygen, getting out carbon dioxide and water here. And we can see that we're going to release some of this energy as heat, but most of the energy that we get from breaking the bonds in the glucose is going to go into this conversion of ADP, or adenosine diphosphate, into ATP, adenosine triphosphate. So remember that those were two of our nucleotides that we talked about. And then we can use ATP. We can use breaking it down to do work in our cells. So we can do work in the form of cellular movement. We can do work in the form of making different molecules, or we can do work in the form of moving different compounds across our membranes. There are lots of different ways we can use this ATP. Okay, so we're gonna focus in on ATP for a second, because this is the key to what's going on. So ATP is our method of moving energy around in cells, and it's all about these phosphate groups at the end. So ATP, basically a nucleotide, we have this adenosine part, and then ATP, our form that has a bunch of energy in it, has these three phosphate groups. And the energy we're thinking about is stored in these bonds at the end. So we basically have energy here in this bond between these last two phosphate groups. So in order to make ATP, we need to put in energy in order to create this bond. And then in order to use ATP, we break this bond and release the energy contained in it. So we're going to see both of those things happening in aerobic respiration. We're going to see breaking this bond to release a little bit of energy. There are parts of cellular respiration that require energy. And we're also going to see formation of this bond, because our main goal is to store the energy that we had in the glucose in this easy to use molecule, ATP. So we're just going to be tracking this phosphate group, popping off and popping back on. Okay, so to synthesize ATP, we need ADP, which is just that same thing, the adenosine plus two phosphate groups bound on covalently. So that's our ADP. It would just have two phosphate groups here. And then if we want to make ATP out of it, we need to add an additional phosphate group. So that's what you see here. This PI would be a phosphate group kind of floating around in the environment. We need energy to put into that bond to covalently bond that phosphate group on. So this is going to give us ATP. We're also going to get some water out of this process. This is an endergonic process, so 7 kilocalories per mole, what we need. It's also a condensation reaction. It's a condensation reaction because we get water out of it as well. We break down ways of making ATP into two sort of buckets. So we can have substrate level phosphorylation, where we basically break a covalently bonded phosphate group off of something else and put it onto ADP to make our ATP. We'll also have oxidative phosphorylation. So in oxidative phosphorylation, we have like kind of some phosphate groups hanging around, And we're going to use something called our electron transport chain in order to bind these phosphate groups on. So in substrate level phosphorylation, we'll see a bunch of enzymes. In oxidative phosphorylation, we're going to see this electron transport kind of cascade. We'll see that works kind of differently, and it's going to depend on chemical gradients across membranes. Okay, so as we're going through the different steps of aerobic respiration, you should be thinking about whether we're making that ATP through substrate level phosphorylation or through oxidative phosphorylation. Okay, so ATP temporarily stores energy. It's easy to break off that phosphate group and get the energy out. This is a hydrolysis reaction, right? Hydrolysis, breaking using water. So when we eventually go to use ATP, we're going to break it down with water. Okay. If we wanted to write in the parts of this equation where we are creating ATP specifically, if we want to get ATP out at the end, we could write in what we use to make it. So you'll see different amounts of ATP written into this reaction. We can make up to 38 molecules of ATP. This will vary a little bit on what tissue type we're talking about, what kind of cell we're talking about. Varies some organism to organism, so don't get too hung up on this number specifically. But we'll see in order to make ATP at the end, we need to put in the same amount of ADP, adenosine diphosphate, and we're going to need to put in some phosphate groups. So you're not usually going to see this written out on the aerobic respiration reaction because we basically cancel them out, but this is where they would go. So remember, in order to get ATP, you need to use up some ADP. Okay. So we could do math about how this all works. So the energy in those 38 moles of ATP is about 266 kilocalories. So that's how much we're getting out of this whole process. And we can release energy during glucose oxidation. We do release energy during glucose oxidation. So we'll see that we release actually more. And this is because we need to use up some energy in the process as well. We can calculate the efficiency of our conversion of glucose into ATP by comparing these two numbers. So here we see the energy that we get out at the end in our form of ATP is 266 kilocalories, but the amount of energy stored was 686. So we can see that we have an efficiency here of about 38%. Now, that might be surprising. You might think that all that energy should go into ATP, but we have to remember that we also have this heat coming off. So we are losing energy in the form of heat as we run this process. So this is going to turn into body heat, some of it, but also just in general, heat in our cells. Okay. Now, the actual steps. Got a couple minutes. Okay. So our stages that we're thinking about are glycolysis, the Krebs cycle, and then oxidative phosphorylation using the electron transport chain. So we can think of those terms kind of equivalently. One is referring to what we're doing. Oxidative phosphorylation is referring to the fact that we're making ATP. The electron transport chain is talking about how we're making that ATP. So what like cellular structure makes the ATP, but they're basically happening kind of at the same time. So if you see these two terms and are kind of confused, like why you see one and the other, it's really sort of emphasizing different parts. Okay, so during glycolysis and the Krebs cycle, which you might also have seen called the citric acid cycle or the TCA cycle, those are all the same thing. We're doing substrate level phosphorylation. So you'll see the phosphate covalently bonded onto something else, and we'll see an enzyme ripping it off and putting it onto ADP. Then we'll have ATP at the end. In the electron transport chain, we'll see that we're going to do oxidative phosphorylation. So we'll just see that it's a different type of process. And since we're going to take a look at what these processes actually look like, that's all we're going to say about that for now. Okay. So the first step of aerobic respiration is glycolysis. Broadly, things to know about glycolysis are that it doesn't produce that much ATP on its own, only produces 2 molecules of ATP on net, but it's really important in your cells because it doesn't require oxygen and it happens outside the mitochondria. Now that's important because we are going to talk about cells that are in high oxygen environments, and we're going to talk about cells that are in low oxygen environments, so cells that have already used up all their oxygen. So it's important to remember that any cell basically can run glycolysis. So if you have glucose, you can do glycolysis. So that's its key piece of importance here. But it is the first step of the pathway generally. So glycolysis starts with glucose. So we get glucose, our monosaccharide, and we're going to break down that glucose molecule into 2 molecules of pyruvate. There are 10 enzyme catalyzed reactions that do this. They all take place in the cytosol. So we'll see that metabolism involves the mitochondria, but not yet. So right now we're in the cytosol, we're just floating around in the cell. We get 2 molecules of ATP out. Important to sort of dog tag in your mind for later is that we are also going to have our sort of helpers, our electron carriers, NAD and NADH. So we're going to see that NAD molecules are reduced to NADH in the process of glycolysis. Now, this doesn't matter to us yet, but this will matter to us later on in the process. We'll see these again when we talk about the electron transport chain and oxidative phosphorylation. Okay. So if we wanted to write out an equation for what's happening in just glycolysis, it would look different than our equation for all of aerobic respiration. So we'd see that we have glucose, we add in 2 molecules of NAD, we add in 2 molecules of ADP, 2 phosphate groups. We get out 2 molecules of pyruvate, 2 molecules of NADH. So we reduced our NAD. We have 2 free-floating hydrogen ions, and we have 2 molecules of ATP. Okay. Don't focus too much on specific numbers. I know these are going to be a lot of steps, a lot of things you're kind of separating in your mind. What I want you to focus on as you're thinking about like these other components like the NAD and NADH, I want you to think about relative amounts. The only thing that you really have to remember the numbers of is basically the ATP here in this process. I do want you to know that we make 2 ATP during glycolysis, but the rest of these numbers around aren't that important, except for the fact, sorry, I'm gonna make another exception now. We'll see that at this point, once we split glucose into 2 pyruvate molecules, we've split a molecule in half. So everything further from this step is running in parallel, basically. Okay. I want to talk for a second more, focus in on NAD and NADH. So when we were talking about nucleic acids, we did mention these. We mentioned that they're energy transferring nucleotides. So they are nucleotides. They're similar in structure almost to ATP. They're also coenzymes. So they're not going to catalyze specifically any processes here in aerobic respiration, but they are crucial to the function of some of our enzymes at the end. So NADH is the reduced form of NAD. So we reduce NAD, which adds an extra hydrogen onto the molecule, and this form has energy in it. basically. So NADH has an extra electron, so it has kind of extra energy in it. So this is going to be carrying around energy from different places, like from glycolysis, to the end of our process when we're eventually going to make a ton of ATP. If we wanted to look at all the steps of glycolysis, it would look like this. You don't need to memorize all these enzymes and intermediate steps. But we do want to think about this as having kind of two conceptual parts. So we have an energy investment phase at the beginning of glycolysis, where we actually have to put in 2 ATP molecules. So it seems kind of counterintuitive that we're using ATP in the process of making ATP, but it's true. At the beginning of this process, we need to add in some energy in the form of ATP in order to basically destabilize the structure of this glucose molecule in order to allow us to split it into our pyruvate molecules. So in our energy investment phase, we use ATP, but we then also have an energy harvesting phase. So after we destabilize this molecule and split it in half, we are then able to get out some ATP molecules, and we get out more than we put in, which is why we have a net yield of 2 ATP. Because we spent 2 ATP, but we earned 4 ATP. So overall, we made ATP. So you can see here, you don't need to know the names of these specific enzymes, but you can see that here where we make ATP, we used a kinase. Here at the end, we use another kinase to make more ATP. So that goes back basically to understanding tacking on phosphate groups, right? We talked about that when we were talking about covalent regulation of enzymes. This is also how we're making the ATP using a kinase. So this is substrate level phosphorylation, basically is what I'm saying. Substrate level, we're thinking about enzymes. This is another big picture of the process. You don't have to memorize this whole process, so I'm not gonna spend a ton of time on it. Said this a couple times, so I'm not gonna put this up as a poll right now, but I'm gonna give you a couple seconds. Hammer home this message: glycolysis generates ATP using, you're all gonna answer like on your piece of paper, and I'm gonna tell you the answer. Yay, hopefully we all answered that it's substrate level phosphorylation. Okay, so we're going to end there for now. On Monday, we'll talk about what happens to that pyruvate. We'll talk about the rest of aerobic respiration, and hopefully we'll get onto a short little case study to make sure that we kind of understand how this process works. So take a look at your old chemistry notes on metabolism so that you're ready to go on Monday, and don't forget to do the quiz.